Hello, this is Dr. Beall again. Today I will be talking to you about signal transduction. Here's an outline of the presentation. First, I will present briefly what signal transduction is and why it is important in biology and especially in dentistry. Second, I will talk about general aspects of signal transduction and cell signaling. And after that, I will talk about receptors, including three important families that come up again and again. G-protein coupled or 7-transmembrane domain receptors, tyrosine kinase receptors, and nuclear hormone receptors, which I also mentioned in the lecture on gene expression. Next, I will talk about various signal transduction pathways and second messengers, including cyclic nucleotides, inositol lipids, calcium ions, MAP kinase, and NF-kappa B. And lastly, I will go through some examples of cell signaling and signal transduction in the oral cavity, taste signaling in taste buds, and control of saliva flow by salivary glands. We will see how some of the molecules and pathways we look at generally are doing functions in the oral cavity. What is involved in signal transduction? For cells to fulfill their functions in the body, they often need to make signals and respond to signals. Those signals can be between cells, and there are different kinds of signals that may not be totally exclusive categories. Hormones are one type. Generally, there's something that spreads through the whole body, like sex hormones, growth hormone, stress hormones, and so on. Neurotransmitters generally communicate from one neuron to another, and often only in the tiny region of the synapse. The derivation of the word cytokine means generally a signal between cells, but the term is almost always used for signals that are either sent or received by immune cells in particular. And growth factors are signals that cause recipient cells to divide and grow, as well as other effects. There are also signals that cells can get from the environment. These could be the level of oxygen, the presence of molecules like glucose, and we saw examples of those kinds of things in the gene expression lecture, or the presence of foreign organisms like bacteria or viruses, or the various stimuli that are detected by sensory organs, such as light, taste, or odorants. The general paradigm is that there is information outside of the cell, and it can cause changes inside the cell, and that can lead to the final effect. And that's the process that is called signal transduction. Why would dentists need to know about signal transduction? One reason is that many of the processes that go on in the mouth, either normally or during or after dental procedures, involve signal transduction. Examples are wound healing and blood clotting, and there are dental products on the market that contain growth factors to stimulate healing, for instance. Pain also involves signal transduction, and obviously that's a big concern in dentistry, at least to the patients. Lastly, cancer of all types, including oral cancer, involves signal transduction. I'll show examples in this lecture of signal transduction genes that can be turned into oncogenes by mutations. Another reason why signal transduction is important to clinicians is that many drug mechanisms act on signal transduction pathways. There are two major ways that they do this. One is by acting on receptors. Agents that block receptors are called antagonists, and agents that stimulate receptors are called agonists. Those are the pharmacological terms for different agents. A second major way is to inhibit transduction steps after the receptors. So for instance, there are drugs that can inhibit kinases that transduce the signals, or aspirin that inhibits the cyclooxygenase enzyme that's involved in creating a signal. Now I'll show you some general aspects of signal transduction. Here's a general pathway of cell signaling. It starts with an extracellular signaling molecule that binds to a receptor protein. Then the signal moves through the various intracellular signaling proteins, and eventually the signal reaches the target proteins, which mediate the cell's response. For example, it could affect a metabolic enzyme to change metabolism, a gene regulatory protein to cause altered gene expression, and we saw some examples of that in the gene expression lecture. Or it could affect a cytoskeletal protein to cause altered cell shape or movement. And really, muscle contraction is just an extreme example of a cytoskeleton-mediated effect, where the neuron sends a signal to the muscle cell and causes, contraction of the entire cell that can, move limbs and so forth on a broad scale. And we'll talk a little bit about muscle contraction when we get into the effects of calcium in signaling. Receptors for signals can be located either on the cell surface, which is true of most receptors, because most signaling molecules are hydrophilic, as seen here. So that in that case, the signaling molecule is soluble in aqueous solution, so it can move in the blood plasma or in the intracellular space. Another possibility that's somewhat rarer is to have small hydrophobic signaling molecules that can pass through the cell membrane and bind to an intracellular receptor that can go directly into the cell nucleus. In this case, the signaling molecule is not highly soluble because it's hydrophobic, so it is likely to be associated with a carrier protein to get it transported between cells. And the main examples of this type of signaling are steroid hormone-like and similar receptors. Now we'll discuss some of the different modalities of intracellular signaling and some of the terminology. It is possible for a cell to signal through molecules that are membrane-bound, and that is called contact-dependent signaling. An example of this type is in the immune system between antigen-presenting cells and lymphocytes that is key to the acquired immunity. Paracrine signaling is when the signal is produced by one cell and diffuses to other cells that respond to it. Synaptic signaling is done by neurons, where they release a neurotransmitter in the tiny region of the synapse to affect the target cell. While endocrine signaling involves the production of hormones by endocrine cells, which circulate in the bloodstream and can affect target cells at great distances. Finally, autocrine signaling is where a cell releases a signal that can affect itself. It's contrasted to paracrine signaling. The example in the slide shows that a group of cells can amplify an autocrine signal over a single cell. Interestingly, bacteria have been shown to do this exact process, and in bacteria it's called quorum sensing. Then they will change their gene expression based on how many of their species are in the close environment. Those were some general aspects of signal transduction and its terminology. Now I will move on to discuss receptors. The first type of receptor that I will talk about is called either of two names. The two names are G-protein coupled receptors and 7-transmembrane domain receptors. Here are some basic facts about them. They have many important effects on health, so much that over 1/3 of prescription drugs are acting on this type of receptor. As implied by the name, they have a structure with 7 transmembrane alpha helices. Finally, they can be activated in a variety of ways and lead to a variety of effects. Most often they are bound by a signaling molecule, but there are cases like rhodopsin, where the light photon changes the structure of a retinal molecule that is covalently bound to the opsin protein, and the opsin protein is a 7 transmembrane domain receptor, or the thrombin receptor, which responds to being proteolytically cleaved by the thrombin enzyme, and the thrombin receptor is also a 7 transmembrane domain receptor. Other effectors can include molecules that you smell or taste, small molecules like epinephrine and serotonin, or larger peptides like hormones and cytokines, and many more. There are thousands of these receptors in our genome. This shows a cartoon of the structure of the receptor. And the amino acids are shown as the dots, and we can see the amino terminus of the protein is extracellular, and then it has the 7 alpha helices that cross the membrane, the 7 transmembrane helices, and the carboxy terminus then is intercellular. The numbered dots are various amino acids that are mutated in human genetic diseases in different receptors, and I won't get into that level of detail now. In the last slide, you could see why these receptors are called 7 transmembrane, or 7TM would be the short version of that. This slide shows why they're also called G-protein coupled receptors, or GPCRs, is the short version of that. Here's the receptor embedded in the plasma membrane. In this case, it is somewhat more realistic three-dimensional view than the last one slide, and you can see that the transmembrane helices gather into a bundle in 3 dimensions. The ligand binds to the extracellular part of the receptor, and that then is connected to a protein called the communicated to a protein called the heterotrimeric G protein, as seen here. The heterotrimeric label tells you that it has 3 different protein subunits: alpha, beta, and gamma. The alpha subunit binds to GTP or GDP, which is where the G protein part of the name comes in. Again, this is the same G nucleotide that is a building block of RNA. These nucleotides like ATP and GTP pop up in all kinds of processes in the cell. Another thing that is not labeled on the figure is that the alpha subunit and the gamma subunit are modified by covalent addition of lipids, which are shown as these black squiggly lines. These lipids insert into the membrane bilayer by hydrophobic interactions. So the G proteins are kept at the inner surface of the cell membrane, which is where they need to be for their function. The last part of this diagram shows that the G protein then in turn interacts with an effector protein that produces an intracellular second messenger, and that transmits the signal to the rest of the cell. This diagram walks through the steps in G-protein coupled receptor signaling, with the molecules colored the same as the previous slide. First, the receptor is unbound, and the G protein has GDP bound. Then the ligand binds to the receptor and changes its structure in a way that allows binding to the G alpha. That in turn causes GTP to replace GDP on the G protein, on the G alpha subunit in particular. Then the alpha subunit dissociates from the beta gamma subunits and the receptor and binds to the effector. That stimulates the effector. In this case, it's adenylate cyclase, so it converts ATP to cyclic AMP, which is a common second messenger. At some point, the alpha subunit acts as a GTPase and converts GTP back to GDP. The alpha protein then stops activating the effector and goes back and can rebind to the beta and gamma subunits. The last two steps show a process that is called desensitization, and we'll get more into that in some later slide. So to recap these things then, G proteins are on in terms of transducing the signal when GTP is bound to the alpha subunit. That's to say when they're interacting with the effectors. On might be a little bit confusing because some of the G proteins can either stimulate the effector or inhibit the effector, but they only interact with it and carry out their function with the GTP bound, not when GDP is bound. So it's two different states, and one's the GTP form is when it can interact with the effector. The alpha subunit has an intrinsic GTPase activity that converts the GTP to GDP and turns it off in terms of interaction with the effector. And two important effectors are adenylate cyclase and phospholipase C beta, but there are many others as well. And they're in the number of dozens of different G proteins and different alpha and beta subunits and so forth. This slide now shows that process of desensitization that I mentioned earlier. This allows G protein coupled receptors to be able to tune the response so they don't continually respond to a set stimulus. For instance, these receptors are involved in the process of chemotaxis of immune cells. And in that process, they sense bacterial molecules or cytokines and allow these immune cells to migrate along a gradient towards higher concentrations. And that's kind of how the immune cells that are in the blood, sense that there's an area where they're needed, where there's an infection or inflammation, and they need to migrate from the blood into the tissues. But that process requires that the response is tunable to a concentration of ligand, because they migrate towards the higher concentration until they reach the source. So they need to be able to sense all different concentrations along a gradient of different concentrations. And one way that these receptors are able to do processes like that is through this process called desensitization. And it's shown here that an activated G protein coupled receptor stimulates a kinase called GPCR kinase, or GRK, which phosphorylates the internal parts of the receptor and allows the binding of a protein called arrestin. And that arrestin prevents interaction of the receptor with the G protein and can also target the receptor to these clathrin-coated pits, which are a mechanism that the cell uses to remove the cell surface membrane and return it to the inside. So in that case, the receptor is removed from the cell surface because it's targeted to these clathrin-coated pits. Now I will move on to receptor tyrosine kinases. These receptors act through the reaction seen here. You hopefully know that a kinase is a type of enzyme that adds phosphate to a molecule, though the name is not very descriptive. Kinases are pretty prevalent in signal transduction, especially protein kinases. And tyrosine kinases are one type of protein kinase. Here we see a tyrosine residue in a protein where the peptide chain extends off to the left and right. And the tyrosine kinase transfers phosphate from ATP to the hydroxyl group of the of the tyrosine. Receptor tyrosine kinases tend to have the structure shown, with a single transmembrane domain, an extracellular domain that binds the ligand, and an intracellular domain that carries out the kinase reaction. I'm going to digress slightly because the concept of oncogenes is going to come up, and I just want to make sure we know or remember what those terms mean. So oncogenes are genes that are mutant in human tumors and can be transferred to non-tumor cells and make them tumorigenic. It's been known for a long time that some viruses carry tumors, can cause tumors, and this is because they contain oncogenes. So many oncogenes were originally found in viruses, but they often have normal forms in the genome as well. These normal forms of genes that can be mutated are called proto-oncogenes. Finally, there are genes that are inactivated in tumors and when inactivated cause tumorigenesis, and those are called tumor suppressors, which I'm just mentioning briefly for completeness because I don't believe those will come up that much, but mostly we're going to, a lot of things we're going to involve oncogenes. So to go back into receptor tyrosine kinases, we've seen there the proteins that tend to have a single transmembrane domain and a ligand binding and a kinase domain internally to the cell. And when the mechanism was looked at in more detail, it found they didn't actually act through a single subunit. But the mechanism almost always involved dimerization of the receptors. And there are two major mechanisms that are found, ligand-mediated and receptor-mediated. So in ligand-mediated dimerization, each receptor monomer binds to a different part of the ligand, and this causes it to dimerize. While in receptor-mediated dimerization, each monomer binds a separate ligand, and that changes the conformation of the receptor, leading to dimerization. Once dimerization has occurred, the process is similar in either case. The tyrosine kinase domain of each of the receptors acts on the other monomer in the dimer, which is called trans-autophosphorylation. So they're putting phosphates on the tyrosine of the other monomer in each case. After that, the presence of phosphotyrosine on the intracellular domains is sensed by binding proteins. The signaling proteins that bind to phosphotyrosine have one of two classes of protein domain that do this function. These are called SH2 and PTB domains. This shows the major families of tyrosine kinase receptor with representations of the various protein domains that are present. The extracellular ligand binding part is very diverse from family to family, with domains that resemble other proteins. For instance, the blue cylinders are similar to immunoglobulin, but the intracellular part is more constant from family to family and includes the tyrosine kinase domain in red. I'll just to show one family here. That family includes the insulin receptor. So actually, insulin, the hormone, is acting through a receptor tyrosine kinase. Others are receptors for various growth factors. If you see GFR ending it means it's growth factor receptor. For, say, FGFR is fibroblast growth factor. VEGFR is vascular endothelial growth factor receptor, and so on and so forth. And those growth factors are often important in cancer and in wound healing. Once the receptor has undergone trans-autophosphorylation, there are several ways that signaling can proceed. One pathway that we're going to go through in more detail involves an adapter protein called GRB2 that contains the SH2 which is the tyrosine kinase binding domain that I mentioned, and it's short for Sarc homology 2. And that binds to the phosphotyrosine, which in turn the GRB2 also binds a protein called son of sevenless, or SOS, which then activates a protein called RAS. And I'll talk more about RAS and a little bit about GRB2 shortly. A second possible pathway involves a scaffolding protein called IRS that has the PTB domain that I mentioned is the other kind of phosphotyrosine binding domain, not the SH2. IRS in turn can be phosphorylated on tyrosines by the receptor along that tail part of it, and then it can bind additional proteins. And there's shown a couple additional proteins bound there. In the third example, there's a related mechanism that occurs with transcription factors in the STAT family. In this case, the tyrosine kinase that acts on the STAT factor is actually a separate protein from the receptor, but the STAT protein does get phosphorylated on tyrosine and also contains SH2 domains, so it can bind the phosphotyrosine on the other monomer and form a dimer. And that dimer gets translocated into the nucleus and turns genes on and off. Then as a fourth mechanism, there can be also effector proteins that bind directly to the receptors. So shown here are a few of those, and I'll discuss the roles of PI3K and PI-PLC gamma a bit more when I go into inositol signaling. A major point of these different pathways is that the SH2 domain is present in a large number of signaling proteins, and that its role is to bind to phosphotyrosine. So that SHP2 protein, the PI3K, the PI-PLC gamma, they all have SH2 domains, as does the STAT, as does the GRB2. So SH2 is a very common motif and involved in binding to phosphotyrosine. Now I'm going to discuss the RAS protein that was in the first part of a previous slide, that's getting signals from receptor tyrosine kinases through the GRB2 and SOS proteins. So RAS was originally found in these it is a proto-oncogene, and the oncogene form was originally found in a couple of viruses called the Harvey and Kirsten sarcoma viruses. And they cause sarcomas in rats. And RAS, the name, is short for rat sarcoma. Later, it was found that the viruses had gotten the genes from the host genome, and there are in fact 3 RAS genes. So HRAS is most like the gene in the Harvey virus, KRAS is most like the gene in the Kirsten virus, and there's a gene called NRAS that was first found in neuroblastoma cells. So that's why it got the name N. So, and it was found that one or the other of the human counterparts of these 3 genes are mutated in human tumors. And in fact, mutated RAS genes are the most common oncogenes, and they're found in about 20 to 25% of cancers. So there's a lot of different genes that can be potentially oncogenes, but RAS is the most common one. The 3 RAS genes are part of a family of proteins called the small GTPase family that has a number of similarities to the heterotrimeric G proteins that interact with the 7 transmembrane receptors that I talked about earlier. Now, 2 differences are that there's only a single subunit instead of 3 subunits as in the heterotrimeric family, and they're much smaller in size. And three similarities of RAS to the other G protein family is that there are two forms with either GTP or GDP bound. They have GTPase activity, so they can convert the GTP to GDP, and they are modified by covalent attachment of lipids to localize to the inner surface of the plasma membrane. And as in with the alpha subunit of the heterotrimeric G proteins, the GTP bound form is the active form and the GDP bound form is the inactive form. Here's the structure of RAS and the cycle that RAS undergoes during signaling. Looking at the structure, this superimposes the structures of RAS with GTP, that's in red, and RAS with GDP in green. And you can see changes that occur in the structure in red and green, especially in the two regions labeled switch 1 and switch 2. Those changes then allow it to bind and activate effector proteins in the GTP form. The left-hand side then shows the GTPase cycle. This example starts with the inactive small GTPase bound to GDP. One thing that can occur is that the protein interacts with a protein called a guanine nucleotide dissociation inhibitor, or GDI, and that can lock it into an inactive state because it needs the GDP to be exchanged for GTP to become active. That activation pathway is shown across the top of the diagram, and it's stimulated by a protein called guanine nucleotide exchange factor, or GEF. That binds to the GTPase and stimulates GTP exchanging for the GDP, which switches on the GTPase. In the example of 2 slides ago, the GEF function was carried out by the son of Sevillus, or SOS protein. Next in the cycle, the GTPase activates the target proteins and moves the signal along downstream. And the GEF comes off during that process. Then in the last part of the cycle, a GTPase activating protein, or GAP, comes in. As the name suggests, this stimulates the GTPase that's to convert the bound GTP to GDP, and this inactivates the signaling. And the diagram shows a clock because depending on how active the GAPs, how actively the GAP stimulates GTPase depends on how long the response stays active. At any rate, once the protein has GDP bound again, it can then start the cycle again and become active. Here's the ribbon structure of the GRB2 protein that was shown earlier, being an adapter between the receptor tyrosine kinases and SOS, or son of Sevillus. And we can see clearly what's going on with the SH domains. And we're introducing a new domain that's SH3 here. You can see that the GRB2 has an N-terminal SH3 domain, an SH2 domain in the middle, and a C-terminal SH3 domain. And you can clearly see how these domains are really independent structural units, and why these domains are used over and over again, because you can sort of plug them into these structures and mix and match things. SH in the SH2 and SH3 stands for SRC homology, or it's because these domains were originally found in an oncogene from Rouse sarcoma virus. And even though the gene name is spelled SRC, it's always pronounced SARC in my experience, because it's short for sarcoma. That oncogene that's from that virus is actually a non-receptor tyrosine kinase, and that protein has 3 domains. The kinase domain, along with the original gangster SH2 and SH3 domains, as you will. So the 3 domains are the tyrosine kinase in that molecule, and SH2 and SH3, but other proteins only have SH2 or only have SH3 or whatever. It's just that SARC was where they were originally found. The SH2 domain, as I told you, binds to phosphotyrosine, which is on the receptor side, and SH3 binds to the SOS protein in this case. So that's how it can adapt and bridge between the receptor and the SOS protein, which is going to go on and be the GEF factor, the guanine nucleotide exchange factor, that will activate the RAS, and RAS will go on to activate the effectors. Moving on to nuclear hormone receptors. These are responsible for signaling from a number of compounds, some of which are shown here. At the top here are sex hormones, and of course there are various ways that receptors for those are manipulated pharmacologically, for birth control, to treat breast cancer, for gender reassignment, and hormone replacement. In green are another group of hormones that are derived from cholesterol, the corticosteroids. And cortisol or hydrocortisone is an important drug because of its anti-inflammatory effects, while aldosterone affects kidney function. Thyroid hormones are made by the thyroid gland and affect metabolism, and they are used to treat hypothyroidism. Vitamin D is another compound that works through nuclear hormone receptors, and it's involved in calcium homeostasis and metabolism. And retinoic acid has effects on intercellular signaling during embryonic development. Therapeutically, it's used to treat acne, but of course it's contraindicated to use during pregnancy because of the effects on embryos. There's a few other ligands that are known for these nuclear hormone receptors but not shown, and what they all have in common is that they're quite hydrophobic molecules. And that allows them to use the mechanism that's shown in this panel, where the hormone can penetrate through the plasma membrane on its own and then bind to the receptor. In the case pictured, it's binding in the nucleus, although for some receptors the protein is in the cytoplasm and translocates into the nucleus after binding the ligand. The binding changes the structure of the receptor and allows it to bind to specific sequences in genomic DNA and regulate gene expression directly. The receptors have a zinc finger type of DNA binding domain that I mentioned in the lecture on gene expression, and they are considered to be transcription factors as discussed in that lecture. So they're combined a receptor and a transcription factor. Basically, then, the mechanism of action of nuclear hormone receptors is fairly simple. They bind to the ligand, and that changes their structure, and then they can bind to DNA and change gene expression. Here's a couple further aspects of their function. There are different types that behave differently with respect to whether, as I mentioned, whether they are in the nucleus or cytoplasm when the ligand is not there, and some other properties. And so in the ones for retinoic acid and thyroid hormone, for example, stay in the nucleus. The steroid ones are in the cytoplasm, and then when the ligand binds to them, they translocate into the nucleus. They can have other proteins that work with them and are called coactivators and co-repressors. And sometimes these proteins have effects on on chromatin remodeling, which is a subject that I covered in the lecture on gene expression. So the co-activators are sometimes histone acetylases, and the co-repressors are sometimes histone deacetylases, as we heard about in that lecture. And tamoxifen is an estrogen receptor antagonist, so a blocker of the estrogen receptor, and it's used to treat breast cancer. And it appears to block the co-activator binding to the receptor. So that's an interesting case. And lastly, there are quite a few of these receptors in the genome, and some of them we still don't know the ligand, and we call those orphan receptors. Those are some of the major receptor types, those three that I talked about, but there are also many others, and I'll briefly mention some of them. There's a group of receptors called the cytokine receptor family that, unlike the receptor tyrosine kinases that I talked about, do not have intrinsic kinase activity, but instead associate with intracellular kinases. These include receptors for interferon and growth hormones. One of the kinases that they associate with is called JAK, J-A-K, either for Janus kinase or just another kinase, depending on what you read. JAK kinase frequently phosphorylates that STAT protein that I mentioned earlier, that is a transcription factor that when phosphorylated goes to the nucleus. So then that's called the JAK-STAT system. There are receptors that have kinase activity that work on serine and threonine in proteins rather than tyrosine. Example of that is the receptor for the transforming growth factor beta. Also, there are receptors that are ion channels, so that the ligand directly affects the permeability of the membrane to ions. And an example of this type is the nicotinic acetylcholine receptor. Note that the muscarinic acetylcholine receptor is totally different and is one of the G protein coupled receptor family. That's the first type that I mentioned a while back. Finally, since I know you're really wanting to hear more about kinases, there are histidine protein kinases that are the receptor part of a common regulatory mechanism called two-component systems and are found mainly in bacteria. Now I will talk about second messengers and pathways. We've seen a number of different kinds of receptors and directly associated molecules. And in some cases, like the nuclear receptors or ion channels, that comes fairly close to being the whole story, and the receptor can pretty much cause an effect on the entire cell. But in other cases, it's useful to amplify the signal in terms of number of molecules involved, to spread it through a wider volume, and that often involves second messengers or signaling pathways. Today I'm going to discuss a few of the more common systems. The first one will be the cyclic nucleotides, cyclic AMP and cyclic GMP. Next will be inositol lipid derivatives and other lipid second messengers. Then I will talk about calcium ions. Then I will talk about a couple of pathways. The MAP kinase pathway, because I have the feeling you really want to hear more about kinases. And then the NF-kappa B pathway. So here's cyclic nucleotides. This is the reaction that converts ATP on the left into cyclic AMP on the right. I'll mention again that biology is repurposing the RNA nucleotide building blocks, the ribonucleotides, for signal transduction, as it often does. The enzyme is called adenylate cyclase, and it kicks out pyrophosphate and forms a bond from the remaining single phosphate in the 5' position to the 3' position, so it's now cyclic. That bond can in turn be broken by an enzyme called phosphodiesterase to form AMP, adenosine monophosphate. Just a reminder that this was mentioned in passing in my previous lecture, that E. coli makes cyclic AMP when it doesn't have enough glucose, and that is bound to the catabolite activator protein as the positive control part of lactose regulation. Cyclases and phosphodiesterases can be the targets for activation or inhibition by G proteins or small GTPases, as we have seen. There are a few additional points about cyclic nucleotides. One is that cyclization can occur with guanosine as the base, and in that case you get GTP converting to cyclic GMP, converting to GMP. Cyclic GMP and cyclic UMP, cyclic CMP and cyclic UMP seem to be less important, although there's some evidence that they're also used as cyclic nucleotides. The cyclases for A and G are different. So cyclic AMP is made by a different enzyme than cyclic GMP. So you have guanylate cyclase rather than adenylate cyclase. However, some phosphodiesterases work on either A or G. Lastly, the pathway is often targeted by bacterial toxins and drugs. So for instance, some bacterial toxins are adenylate cyclases, and compounds like sildenafil, which is a generic name for Viagra, works by inhibition of a cyclic GMP phosphodiesterase. So in that case, it's a specific GMP phosphodiesterase, but there also are phosphodiesterases that will do either A or G. Since I haven't mentioned kinases in a while, there is a kinase called protein kinase A that is involved in many responses to cyclic AMP in human cells. If cyclic AMP is not around, the protein kinase A is present as a tetramer with 2 regulatory subunits in red and 2 catalytic subunits in blue. The regulatory subunits inhibit the catalytic subunits in this configuration. If the level of cyclic AMP rises, it can bind to the regulatory subunits and leads them to dissociate from the catalytic subunits, which then become active. The catalytic subunit then phosphorylates target proteins on serine and threonine that can mediate responses. There is an additional wrinkle that protein kinase A phosphorylates the phosphodiesterase as well, which activates it and helps to bring the response back down. So there's some built-in feedback control of the response. And we'll see this again and again in signaling pathways. There's a sort of built-in feedback control that limits the responses. We've seen similar phenomena in G protein coupled receptors and RAS, that these signaling pathways tend to have some feedback. Now I'll talk about phospholipid-derived second messengers. This slide shows some of the structures and reactions involved in generating lipid second messengers. We start at the top with the typical glycerophospholipid that's the major component of membranes. Note that the R group can be variable, as can the fatty acids. These can be cleaved by these phospholipids can be cleaved by various kinds of phospholipases to release various molecules. So phospholipase A can cleave off fatty acids. Phospholipase C cleaves between glycerol and the phosphate to release diacylglycerol and the head group, and that's going to be an important one. And phospholipase D cleaves after the phosphate to release the R group and leave phosphatidic acid. So looking at the phospholipase A, one fatty acid that can be released in this way is arachidonic acid, and that is the precursor for the second messengers called eicosanoids. R groups with inositol are important in signaling, and we'll discuss them further. And so they'd be released. Inositol would be released by the phospholipase C as the other product in this inositol 1,4,5 triphosphate would be released as the other product along with diacylglycerol by phospholipase C. And I'll just mention quickly, sphingolipids are involved in apoptosis pathways as signaling molecules, but I'm not going to discuss them in any detail. So So here's some basics about eicosanoids. As mentioned, the precursors for them are frequently arachidonic acid or other 20-carbon fatty acids. Eicosanoids include three major classes: prostaglandins, thromboxanes, and leukotrienes. And some of the structures are shown here for reference. The enzymes that produce them are called cyclooxygenases and lipoxygenases. And these enzymes that produce these molecules are important drug targets. For instance, aspirin affects the cyclooxygenase, and some asthma drugs as well affect the enzymes that produce eicosanoids. This slide shows the structure of inositol on the left without the phosphates that were in the earlier slide. So it is a sugar alcohol with a 6-carbon ring and hydroxyls on each carbon. The particular form of interest is myo-inositol, which refers to the stereochemistry of the hydroxyl groups with respect to the ring, and that's the main one we're talking about. Inositol sometimes is present in the head group of phospholipids, as I mentioned. In the diagram above, there's a phospholipid which is phosphatidylcholine. Choline is attached to the other side of the phosphate from the glycerol, but inositol can take the position of choline in that molecule, and that be phosphatidylinositides instead of phosphatidylcholine. And here's some phosphatidylinositol on the left, and phosphatidylinositol diphosphate, or PIP2, on the right. PI and PIP2. And they are involved in a pathway. And this diagram shows how the second messengers are generated from the phosphatidylinositides. And we get to talk about kinases more. It starts with phosphatidylinositol, or PI. Then phosphate is added by a kinase at the 4 position in step number 1 in that diagram to make PI4P. Then another phosphate is added by another kinase to make PI45P2 in step 2. Step 3 is the G protein activation by a receptor that I talked about earlier. In step 4, the alpha subunit of the G protein with GTP bound activates phosphatidylinositide phospholipase C beta, or PIPLC beta. That breaks the PI45P2 into 2 parts in step 5. As I showed a few slides back, the 2 parts are diacylglycerol in 6 and inositol 1,4,5 triphosphate, or also known as IP3, in step 7. You can also see the diacylglycerol binds to even another kinase called protein kinase C, while the IP3 interacts with a receptor that is on the smooth endoplasmic reticulum. And it has the effect of releasing calcium ions from the smooth endoplasmic reticulum and raising the calcium concentration of the cytoplasm. You might ask, what do we know about protein kinase C? As I mentioned, it is activated by diacylglycerol. It also requires calcium ions. It is also activated by a group of compounds called 4-bol esters that are plant-derived and had been known for a long time as tumor promoters. That is, they don't cause cancer, but they make carcinogens more potent. And protein kinase C has many, many effects, including in smooth muscle contraction, in metabolism in liver and fat, and in the nervous system. That was a quick look at the inositol pathways. Now I will talk about calcium, and that connects in because the IP3 is releasing calcium from the ER into the cells, although calcium can also come out from other sources. Calcium is normally much lower in the cytoplasm of cells than in extracellular fluid. It's around 100 nanomolar in cytoplasm and around 10,000-fold higher outside the cell. And those concentrations are maintained by channels and pumps. As well as the extracellular fluid, the calcium ion concentration is higher in some internal organelles, especially the endoplasmic reticulum. So there are pumps that use energy to pump the ions into the endoplasmic reticulum as well. Calcium in the cytoplasm can then be raised by opening channels and allowing the ion to diffuse, driven by the concentration gradient, using either the IP3 receptor on the ER or channels in the plasma membrane to allow extracellular calcium to enter. Following the entry of calcium, pumps will reduce the cytoplasmic concentration over time back to a baseline. There are two major kinds of protein domain that bind to calcium ions and allow them to modify protein activity. So ways of sensing the calcium concentration in a cell or in an organelle. One type is called the EF hand. It's shown on the left here. It has two helices. And the N-terminal one begins with a glutamic acid, which is a single letter code E. And the C-terminal one begins with a phenylalanine, which is single letter code F, and the helices are positioned something like the fingers of a hand, and there's a loop between them with 6 amino acids that coordinate with the calcium ion. So that's why it's known as the EF hand, for those 2 amino acids at the beginning of the helices, and then the sort of fanciful imagining of the helices as fingers of the hand. On the right is the second domain that binds calcium. That's the PKC C2 domain. It is found in protein kinase C, which, as we mentioned, requires calcium as well as diacylglycerol and other molecules, and can mediate calcium-dependent membrane binding of these molecules. And that structure is 2 beta sheets that bind to 2 calcium ions, as shown. The blue are the calcium ions. Here are some examples of effector molecules that mediate responses to calcium ions. The most important one probably is calmodulin. It is a protein with 4 EF hands. With calcium bound, you'll be shocked to hear that it stimulates kinases. One kinase that it stimulates is calmodulin-dependent protein kinase, and that was found to be important in learning and memory. A second kinase that calmodulin can stimulate is myosin light chain kinase, and this is how smooth muscle contraction is controlled. So calcium signals through the calmodulin-dependent, or through the myosin light chain kinase, and then that phosphorylates myosin light chain, and that starts the smooth muscle contraction. Skeletal muscle contraction is also controlled by calcium ions, but it's slightly different. In that case, calcium binds to a protein called troponin C that also has 4 EF hands, but instead of using a kinase, it actually moves another protein called troponin I on the thin filaments. Those are the filaments with actin that are in the muscle, and that activates the muscle and allows it to contract. The last example of a calcium sensing important protein is synaptotagmins. These are proteins with 2 of the other type of domain, the C2 domain, and they control synaptic vesicle fusion with the neuronal cell membrane. So they're what causes the synaptic vesicles to fuse with the membrane and can communicate to the other neurons. This diagram shows a phenomenon called calcium-mediated calcium release, as it occurs in cardiac muscle. It involves the various components and pumps that I have been referencing. The way that cardiac muscle controls calcium is that In the first step in the diagram is a voltage-gated calcium channel in the plasma membrane that opens in response to voltage changes due to an action potential. That lets in calcium from the extracellular space. In step 2, this binds to a second type of receptor that is present on the smooth endoplasmic reticulum along with the IP3 receptor. This is the ryanodine receptor, which is named after a compound that it binds. But usually it is sensing the initial elevation of calcium in the cytoplasm causes this ryanodine receptor to open up and let even more calcium. In muscle cells, the smooth ER is quite specialized for this purpose and is called the sarcoplasmic reticulum. In step 3, the ryanodine receptor releases calcium from the endoplasmic reticulum and amplifies the response. Finally, in steps 4 and 5, the calcium is pumped back into the ER to end the response through a calcium pump in the ER membrane and a sodium-calcium exchange mechanism in the plasma membrane. The whole process occurs every heartbeat, which gives you an idea of how fast these calcium fluxes can go. A process that's being actively studied now is called store-operated calcium entry, or SOCE, and that is shown on this slide. Because the ER has a limited volume, if calcium is released repetitively from there, it may get depleted in the ER. What has been observed to happen in that case is that there are proteins called STIM1 in the ER membrane and a channel called ORAI1 in the plasma membrane that are normally randomly distributed, as shown at the top. In the event of calcium depletion, the STIM1 clusters in the ER membrane at positions near the plasma membrane, and ORAI1 clusters opposite it, following which ORAI1 opens and lets calcium into the cell, which can then be picked up by the ER with its pumping system, and you can restore the levels of calcium in the ER. Next, I will present about the mitogen-activated kinase system. I know, again with kinases, MAP kinase pathways involve 3 sequentially activated protein kinases, so 3 for the price of 1. They are present in all eukaryotes and have many different flavors, which you can see in the diagram on the left. There's listed all the different possible MAP kinase pathways. They're also very important in cancer. That's implied by the name because the definition of mitogen is a signal for a cell to divide, and uncontrolled cell division is a hallmark of cancer. So the mitogen-activated kinases are obviously going to be important, and some of them are oncogenes and so forth. That, like, if you see RAF and MOS up there in the diagram, those are known, oncogenes that were originally discovered in viruses, similar to things we've seen. In the diagram on the left, you can see there are 4 different classes of pathway involved, with somewhat similar inputs and outputs, but differences in the genes, certainly in the kinases, and there's always 3 steps of sequential phosphorylation involved. And those are shown there, so those kinases, as well as the specific names of each one, the generic names are just the MAP kinase is the last one, the MAP kinase or MAP2K is the middle one, and the first one is the MAP kinase or MAP3K. Now in the right-hand diagram, it goes through a longer pathway and sort of integrates this with shows how this integrates with things we've learned about already. And at the top there is you have a receptor tyrosine kinase that's binding to a growth factor. Then GRB2 and SOS get involved through remember the SH2 domain of GRB2 binds to the phosphotyrosine of the receptor, and SOS is the guanine nucleotide exchange factor for RAS. And so that happens. RAS exchanges from DP, the inactive form, to GTP, which is the active form. And that causes the MAP kinase, MAP3K, that's called RAF in this case, RAF, to be activated by binding it to the membrane. And then RAF phosphorylates MEK, which is the MAP2K of this particular pathway. MEK phosphorylates ERK, which is the MAP kinase. And ERK then can phosphorylate transcription factors and to favor cell growth. And one of the genes that is turned on is a phosphatase called MKP1, and that dephosphorylates the MAP kinase. So as for other pathways, there's a feedback loop limiting the response. In real life, things can get very complicated, and as a scientist, I've often gone to seminars and see pathways that look kind of like bowls of spaghetti. Don't worry, if you just totally know the pathways shown in this slide, you should be good for the exam. I'm just kidding, by the way. The thing to take away from this is that a lot of what is going on involves the same sorts of units. There are kinases, of course, they're the red in the diagram. Phosphatases are the light green. G proteins and GTPases are purple. Second messenger enzymes are dark green, and transcription factors dark blue. And the other thing to realize is that there's crosstalk between the pathways at multiple levels. So you can see like protein kinase A is getting involved with the MAP kinase pathways, and through G protein coupled receptors and so forth and so forth. So these, things often get very complicated in real life. One way that MAP kinase pathways avoid crosstalk is by having the kinases anchored to scaffolding proteins. I mentioned that MAP kinase pathways are present in all eukaryotes, even yeast. And in yeast, researchers were able to figure out the following, what I'm going to talk about here. So there are two pathways that involve MAP kinases in yeast that are of interest here. One of which is needed for mating. So yes, actually, yeast do mate. That's so now you know there's hanky panky going on in your beer. Then, but there is a G protein coupled receptor for something called mating factor that's part of this process. And that activates a series of 3 kinases that are bound to this scaffolding protein called STE5. And maybe this is obvious, but STE is short for sterile. So, these mutations in these proteins make the yeast unable to mate. And in normal thing, that the 3 kinases, the signal along that it signals the transcription factors and generates a mating response. And a partially different MAP kinase systems works in response to high salt. Maybe this might be when you added salt to your beer or something. In that case, a different receptor is used and a different scaffold called PBS2. In this case, the MAP kinase is actually the same as for the mating pathway. It's this STE11, but the other two kinases are different. The MAP2K is part of the PBS2 scaffold, actually, in this case. So it's a separate domain that is also the scaffold for the other ones. And the MAP kinase, HOG1, phosphorylates transcription factors that cause an osmoregulatory response. What research has found is that, and shown in the third part of this, you could make a chimeric scaffold that had part of the mating scaffold ST5 and part of the osmoregulatory scaffold PBS2, and in that case you could have mating factor that then caused the osmoregulatory response, showing that these scaffolds can be really very important and actually direct the pattern in the pathway of signaling with MAP kinases. Now I'm going to talk about nuclear factor kappa B signaling. The nuclear factor kappa B pathway is conserved from humans to insects, and it's involved in inflammation, immune response, and stress. And it's considered part of the innate immune response. The basic way it works is shown in the diagram. There's actually a small family of NF-kappa Bs, but the one shown is a heterodimer of the proteins RelA and P50. That is in turn bound by an inhibitor called I-kappa B alpha for inhibitor of kappa B alpha, and it's sequestered to the cytoplasm. Stimulation of a receptor signals through a kinase called IKK, which phosphorylates the I-kappa B alpha, the inhibitor, and targets it for degradation in the organelle called the proteasome that does targeted degradation of proteins in eukaryotes. That releases the NF-kappa B to go into the nucleus, the RelA and P50 or the NF-kappa B to go into the nucleus and activate transcription. What kind of signals induce NF-kappa B? Here are some of them. Gram-negative bacteria incorporate a product called lipopolysaccharide that induces this pathway. Or LPS, as it's known for short, is toxic because of this stimulation of immune response, which leads to LPS also being known as endotoxin. Pro-inflammatory cytokines also induce the NF-kappa B pathway, and two examples are tumor necrosis factor alpha and interleukin-1 beta. Reactive oxygen species also induce NF-kappa B, and ionizing radiation as well can induce it. What genes are turned on by NF-kappa B? They include the pro-inflammatory cytokines, including the TNF alpha that I mentioned earlier, as well and interleukin-1 that I mentioned earlier, as well as the interleukin-6. They also induce genes involved in leukocyte recruitment to tissues that are known as chemokines, short for chemoattractant cytokines. And they can induce, the NF-kappa B can induce antiviral proteins like interferons as well to fight off viral infections. So now I'm going to talk about a couple examples of signal transduction in the mouth, specifically, that hopefully you'll see how the general mechanisms that I've talked about can be applicable in these specific systems. The first example of this will be the taste sensation. Their taste buds are located on these structures called papillae on the tongue and elsewhere. So there are 4 different kinds of papillae, and taste buds are located on all of them except for the filiform ones, which have a more structural role. And the taste buds are shown on the foliate papillae there, and then a close-up version of them is shown, and there are 3 different types of cells in there, and the gustatory cells are the ones that contain receptors and detect the taste signal. You probably know that there are 5 main tastes, and here the receptors involves what they're detecting and their structures. So salt receptors detect sodium ions, and they're a channel type receptor. Sour receptors detect hydrogen ions and are also a channel type. Sweet taste receptors are a dimer of 2 G protein coupled receptors and detect sugars primarily. Umami receptors detect amino acids, especially glutamate, and are also a G protein coupled receptor dimer. And lastly, bitter taste is detected by a family of around 30 G protein coupled receptors. And they detect a variety of mostly plant derived compounds, which may be potential toxins. They also have higher sensitivity to the chemical compound than the other taste receptors. The signal transduction varies greatly then depending on the taste. So here's salt compared to sweet. For salt, there's just a channel that lets in sodium ions if they're present in high levels, and that causes membrane depolarization directly. There's also another channel not shown which lets in calcium from the extracellular space, which is needed for neurotransmitter release. For sweet receptors, first the sugar or other sweet substance binds to the receptor. Actually, as I mentioned, a dimer of G protein coupled receptors. It's shown as a monomer here, but it's actually a dimer, a heterodimer. And then through a G protein, phospholipase C is activated, our old friend phospholipase C, which cleaves the PIP2 into, that's phosphatidylinositol diphosphate, into diacylglycerol and IP3. And IP3 stimulates calcium release from the endoplasmic reticulum, as we have seen. And calcium causes membrane depolarization by opening a sodium channel in the membrane, and also is needed for neurotransmitter release to transmit the signal through the nervous system. Now I'll compare the taste transduction to other sensory systems briefly here. So vision is also a G protein coupled receptor, which consists of the protein, the part called the opsin, bound to covalently bound to a molecule related to vitamin A called retinal, that together is called the rhodopsin. And then the transduction from the rhodopsin is through a G protein signaling to a cyclic GMP phosphodiesterase signaling to ion channels. Olfaction or smell is also the receptors are G protein coupled receptors. There's a large family of olfactory receptors, and they signal through G proteins to adenylate cyclase to ion channels. And finally, hearing is a cation channel that responds directly to the mechanical force. So more like the salt and sour taste, but responding to actually opening the channel by the mechanical force. Finally, I will note that kind of a hot area of activity lately has been in looking at capsaicin and wasabi receptors. So capsaicin is the compound that makes chilies hot, and it affects a channel type receptor called transient receptor potential cation channel V1 that also seems to be responsible for sensing heat and pain, whereas wasabi affects a channel called transient receptor potential cation channel A1, which appears to sense cold and pain. So these are things that are, that we can taste, but also seem to be related to the body sensation of noxious temperatures and pain related to that. So those receptors are getting a lot of interest now. Okay, moving on from taste sensation, I'm just going to go into signal transduction in the mouth relating to salivary fluid secretion. So here's some facts about saliva. I was surprised to read that humans typically produce over 1 liter per day of saliva. It seems like a lot. That amount is greatly increased while feeding. So it's thought that 80 or 90% of the saliva produced is happening while you're eating, and there's almost none produced while sleeping. So obviously the production of saliva, is regulated up and down to a great degree. And functions of saliva are, hydration of the food and of the tissues, and initiation of digestion of the food. And there's some antimicrobial defense compounds in saliva as well. So it has multiple functions. And the idea of controlling the flow of salivary fluid, of course, reminds you of Pavlov and his famous experiments. I've looked into his work a bit. Interestingly, there are at least 2 of his dogs that have been taxidermied in museums, and as they were prepared for the experiments, you can see them in those 2 pictures, and you can see that he was collecting for the one. He was, maybe for both of them actually, he was collecting the stomach secretions as well as the salivary secretions. I also found he didn't use a bell. He used other sounds like a ticking metronome. It's set to a specific speed to induce the production of the secretions. So saliva is produced by the acinar cells of the salivary gland. And the acini are the sacs of cells that are at the ends of the ducts that lead out into the mouth. And the saliva goes down the duct is made in the acinus and then moves out down the duct into the mouth. And this is a diagram of an acinar cell with the pathways that lead to fluid secretion shown. And I'll walk through this. And first I want to point out that this is a polarized epithelial cell. So it has differentiated basal and apical surfaces with different functions and different biochemical compositions. There are tight junctions that seal between cells and keep the lumen of the gland separate from the fluid in the body cavity. The initiating event in fluid secretion is binding of the neurotransmitter ligands to G protein coupled receptors. And there are a few different kinds of receptors that can do this, but they're all the G protein coupled type. The activated G protein stimulates phospholipase C, as we've seen in other systems, and that releases IP3. And here, IP3 acts on the IP3 receptor in the endoplasmic reticulum to release calcium ions into the cytoplasm, as happens in all these systems. And the mechanism for fluid release in the gland is believed to involve chloride ions. So we'll walk through how that happens. When the cell is resting, they accumulate to high levels through a couple of mechanisms, an important part of which is a channel in the basolateral membrane that lets in 1 sodium, 1 potassium, and 2 chloride ions at a time. And the chloride ions build up to a level so that there's a 5 times electrochemical gradient from inside the cell to out. So when internal calcium is elevated, that causes opening of this chloride channel in the apical membrane, and the chloride ions go out into the lumen of the gland. Now, at the same time, there are potassium channels that open up that also stimulated by calcium. Because potassium is higher inside the cell, remember, potassium's high inside the cell and sodium's high outside. So potassium always is happy to go out of the cell, and it does that. And that keeps the cytoplasm from becoming too positively charged because of the chloride leaving, which it might otherwise tend to do. And that if it got too positively charged, that would tend to resist the chloride movement out of the cell. So the potassium's kind of balancing the chloride leaving out. An additional movement of ions is that sodium ions, which are mostly outside of the cell, move along the outside of the cell and actually can penetrate through the tight junctions into the lumen of the gland. And that's driven by the negative charge of the chloride ions as well. So sodium ions are just moving through the tight junctions to get into the lumen as well. And overall, then you get a high concentration of electrolytes in the lumen. And that then makes water tend to leave the cell toward the lumen. And that goes through a water channel called an aquaporin. So you get a mass movement of water from the from the cell out towards the lumen of the duct. And then as the saliva moves along through the duct, most of the electrolytes are actually removed by the duct cells. So the osmolarity of saliva once it gets out into the mouth is actually fairly low. But the electrolytes get removed by the duct cells on the way out. There are a couple of further effects that happen during the process that involve the calcium in the acinar cells. One is that the calcium release tends to occur first at the apical surface of the cell. So on the left is a salivary gland acinar cell, a micrograph of the same cell. The top is phase contrast, and the lower 5 are fluorescent images over time that are imaged with a fluorescent dye that changes fluorescent according to the calcium concentration inside the cell. And it's moving from the top to the bottom. And blue is low concentration of calcium, then green, yellow, and red is higher concentration. And so after the first cell, then the cell is stimulated with the neurotransmitter. And the calcium is elevated first and most at the apical side of the cell. That is because the IP3 receptors are more abundant on the apical side of the ER. And spreading to other regions of the cell depends a lot on calcium entry rather than release from the ER, and appears to use a variation of the store-operated calcium entry, or SOCE, that I mentioned earlier. So on the right, we see there's a protein called STIM1. That's the pink protein in the diagram on the right. And STIM1 is in the ER membrane. And inside the ER, it has one of those EF hand structures that can sense calcium. When it senses there isn't enough calcium inside the ER, it bunches up towards the plasma membrane and interacts with channels in the plasma membrane. Either there's one of two channels that seem to work in these acinar cells. One is the ORAI1 that we heard about earlier, and another one is called TRPC1. And that causes those channels to let those two channels to let additional calcium into the cell, and that raises the calcium throughout the cell, including the more basolateral regions. So then, this is just why we might care about the saliva secretion, because xerostomia is a problem, xerostomia or dry mouth, and it could involve the signaling process. So xerostomia can lead to difficulty swallowing, to caries, to oral lesions, or to candida infections. And the causes of xerostomia can be Sjogren's syndrome, which is an autoimmune disease. Radiation can cause xerostomia, or it can be caused by medications and drugs. And probably in the first two cases, it's just the toxicity to the cells is actually involved, but medications or drugs may be involved in inhibiting the signaling process. So if we understand the signaling, that could be a way to possibly control xerostomia. Now I'll go through and review some of the points that I've told you about in this lecture. So I hope I can try to convince you that signal transduction is important in dentistry and medicine, and it's involved in all these biological processes that you might want to worry about as a dentist. So actions of drugs frequently involve signal transduction. The development, cancer, wound healing, and immunity all involve signal transduction very frequently. And there's a tremendous amount of complexity of known and unknown things, and lots of diagrams that can look like spaghetti. But we're trying to just hopefully get some of the things that occur over and over again and learn what's going on with them. And so we've got some common themes. There are these receptor families. So the first one I told you about was, and a very important one, is G protein coupled receptors. They have 7 transmembrane domains. They interact with the heterotrimeric type of G proteins that have alpha, beta, gamma subunits. Alpha subunit has the GTPase, and the GTP bound form is active. The GDP bound form is inactive. The G protein acts through effectors that can include adenylate cyclase. They can include phospholipase C, can include kinases, phosphodiesterase, et cetera, et cetera. They undergo desensitization with GRK and arrestin, G protein coupled receptors. And we heard about tyrosine kinase receptors, receptor tyrosine kinases. There are many different kinds. The extracellular regions are different, but with the intracellular kinase domain more similar. They're usually activated by dimerization. Dimerization leads to autophosphorylation. They can recruit proteins by interaction of the phosphotyrosine with PTB or SH2 domains that can bind it. And small GTPases like RAS may be involved, which are similar in action to the tripartite G proteins, but smaller in single subunit. Other receptor families, the nuclear hormone receptors, their ligands are membrane permeable. They can be steroid hormones. They can be vitamin D-like compounds. They can be retinoids. They can be thyroid hormones. The nuclear hormone receptors function as transcription factors when the ligand is bound. And finally, I named a few other classes of other receptors, ion channels, non-protein kinase, other protein kinases, and so forth. Then we went on to second messengers and pathways, talked about cyclic nucleotides, especially cyclic AMP, cyclic GMP. They're produced by cyclases, adenylate cyclase and guanylate cyclase. They're removed by phosphodiesterases that may or may not be specific to A or G. The cyclic AMP may act through protein kinase A, or in bacteria, it's this catabolite activator protein. Pathways and second messengers, they're lipid-based. They're various types. The one you keep finding is ones that signal through this phosphatidylinositides, which is PIP2 gets cleaved by phospholipase C, which gives IP3 and diacylglycerol. And diacylglycerol activates protein kinase C, and IP3 causes calcium release from the smooth endoplasmic reticulum and raises the intracellular calcium. And then we get into calcium. It can enter cytoplasm from the extracellular space or from the smooth endoplasmic reticulum. It binds to EF hands or C2 domains on proteins. And I talked about MAP kinase pathways. They have 3 sequentially activated kinases. The RAF-ERK MAP kinase pathway is often mutated in cancer, and MAP kinase pathways in general are related to cancer. And the scaffolding proteins can modify the pathway, as we saw in that yeast example. Then I talked about NF-kappa B. It's a transcription factor. It's activated by proteolysis of the inhibitor, the I-kappa B2 inhibitor, after the phosphorylation by IKK. NF-kappa B is involved in inflammation. More specific, oral signal transduction, the taste, that there are 5 different tastes with different receptors that are either G protein coupled receptors or channels. They signal through the G protein coupled receptors signal through phospholipase C and or membrane depolarization. Then finally, salivary fluid secretion, it's driven by G protein coupled receptors again that respond to neurotransmitters. That phospholipase C leads to calcium release, and store-operated calcium entry is also involved to amplify that. And then water flow is driven by osmosis that follows ions into the lumen of the salivary gland. So that's all I've got for you. Thank you.